Okay, so this is 6th BPRI Crash Course. So I thought to have 2 quick questions before Scott starting. It will not take much time just to check whether how many people have attended so far. So just quickly response to that. It will be like just 2 questions, just click one. So that will help for me to record to the Okay, I will keep that little bit open for a 1 or 2 minutes. So while we're waiting, I would like to introduce our speaker today. So today joining Dr. Scott Geib, a research entomologist at the USDA Pacific Basin Agriculture Research Center, Hilo in Hawaii. As a member of BPRI Genome Group, he works closely with Anna with the genome sequencing effort at the BPRI. Dr. Geib is a graduate from Pennsylvania State University. Today, he's going to talk about locus DNA extraction, library preparation, and contig analysis. So Scott, please go ahead and start. I will end this poll and I will redo it once if we have more participants. Okay, thank you, Scott.
Yeah, thank you so much for the invitation. And yeah, so we're a USDA ARS. ARS is the Agricultural Research Service. We're sort of the basic research arm of the US Department of Agriculture. So if you're not familiar with ARS locations, if you're at Texas A&M, we have a research lab there. And there are about 200 different research sites throughout the nation. Our research unit in Hilo, Hawaii, is specific to tropical pest genetics and molecular biology. And so a lot of our time we spend doing genomic types of things on insects. And so I broadened things today a little bit to just say how to sequence an insect genome. If you have access to the chat, you're more than welcome to say the types of things or the ways that you could see that you might have interests in this space. Like, I want to sequence the genome of whatever, or, you know, I have sequenced the genome, or those kinds of things. Or if you have any questions or ideas or thoughts that maybe we can discuss at the end, or I might see them as we go and kind of pick up on that as well. Yeah, so our lab prepared the locus samples for DNA sequencing on PacBio, as well as sent samples to the Baylor group and performed some of the DNA assembly and things. So I'm going to cover kind of the ways we go through that. I know you had calls with Erez and Olga already, I believe. And so I'm kind of staying out of kind of the higher level scaffolding and assembly and things like that. And I think one of the places where there's a little bit of a black box is, you know, so somebody said I'm supposed to sequence something, and someone's going to actually physically put it on the sequencer for me. But what about how do we prepare this sample? How do we look at the sample, know that it's working well? What are all these new technologies that are out there and those sorts of things? So I'll kind of walk through that a little bit. Oh, sorry. Let me real quick. No, it'll be okay. I'm just going to get an email ring in my ear every time I get a new email. So some of the goals and challenges of doing insect pest genomics. So we're involved in some projects where we're doing arthropod genomic sequencing of insect pests, specifically because we're part of the USDA and we're all about making crops grow better and things like that. Additionally, we have projects on pollinator insects, so things like native honeybee species and things like that. Ideally, you'd like to target chromosome scale diploid assemblies for all of these pests. And this is going to require multiple library types. So I think Olga talked a little bit about how you can go from contig assembly into scaffolding, but there are different library technologies that we use to generate these library types. So like the contig assembly sort of contemporary approach would be to use PacBio hi-fi sequencing, could also come from Illumina, could also come from Oxford Nanopore. There's a lot of different ways you could go about that and sort of what are the pros and cons. There are, on the scaffolding side, you can use Hi-C, which I think is extremely robust and sort of a pretty direct way at getting at this. So this is placing the contigs into the chromosomal context. You could do other things like bio-nano sequencing or bio-nano optical mapping, I guess, or generation of linkage maps, which are some other technologies you could kind of put your contigs into a chromosomal context. So we'd like to target chromosome scale assemblies for all these pest species. So the I5K, or the Arthropod Genomics Sequencing Initiative, is part of the Earth Biogenome Project. And they, a while ago, kind of came up with these quality standards of saying we want all the contigs, or half of the N50 contig size to be 1 megabase, the N50 scaffold size to be 10 megabase. Kind of these are old. We're very well beyond this. And we want chromosome assignment of those contigs within the genome. So, you know, if you're doing a person, a human, a cell culture, whatever, usually these standards are very easy to meet, but sometimes we're doing a mite, a thrips, a tiny little insect, whatever. And so we either have to kind of look at how we approach these things and strike out some of these words, like maybe we can't do a diploid assembly. That means separating the two haplotypes of the genome. That has to be done from a single individual. So if you have a very small sample, you might have to pool individuals, or later I'll talk about some other approaches for doing whole genome sequencing from very small samples. So again, this requires multiple library types, things like PacBio Hi-Fi, scaffolding, but our limitations specifically in the insect world are small physical size and limited DNA materials for various methods. So if you're at Baylor and you go to the core facility, they're going to say, oh, you need X many micrograms or nanograms of DNA to prepare a library. You might take an entire beetle, extract DNA from that, and have 1/10 of the amount of DNA that they're recommending. So what do we do in those cases? We also have an extremely wide diversity of taxa, tissue types, samples, sizes, etc. So it's not always a one approach works for all things. If you're working in the vertebrate space and doing a lot of your samples from blood or something like that, you can kind of standardize things. Here we're dealing with Lepidopteran taxa, so that would be moths and butterflies, have scales on them. Other samples, you're talking about what maybe developmental stage is the most important, and those kinds of things. And so a large portion of our samples fall below what would be typically called standard library prep input amounts. That would be the micrograms. And we're working in the low input. And so that's kind of our normal. So working in the sub-microgram input amount for DNA sequencing. And this is going way back, but when we started this project, we were doing the state-of-the-art was a PacBio Sequel instrument, not even a Sequel 2, where each cell had 1 million sort of sequencing points on it. And we were doing what's called continuous long reads. So these are the lower quality, kind of the old style PacBio reads. Not only did you need higher coverage of these long reads, but the assembly algorithms that assemble these long reads are slower. So things like Falcon and Canoe, which are the assemblers that kind of were developed for assembling CLR reads, would take days or weeks of wall time per genome on a cluster system to assemble a genome. You need a lot of really good DNA, you need long DNA, and you need microgram amounts. You're not shearing the DNA, you're trying to get as long of reads as possible. More recently, oh no, what is going on? All right, there we go. Today, the state of the art is the PacBio Sequel 2 instrument. So here now the cells have 8M or 8 million ZMWs per cell or potential sequencing sites. You might have seen the announcement of what they call their Revio instrument. Each of those cells are going to have 24 million sequencing sites. The type of sequencing technology on these are going to be the same. It's what's this Hi-Fi circular consensus reads. These are reads where you insert, rather than a very long read, you're putting in a somewhat shorter read, so something between 10 and 20 kb in length, but you're creating what's called the barbell library, which is sequenced as a circle, and you're sequencing that 20 kb or 15 kb or 10 kb region over and over and over again. So what you're allowed to do, able to do, is create a circular consensus read, which is what they coin as a Hi-Fi read, where the read consensus accuracy is high. So it's a Q30 read or even higher. Again, there are hi-fi specific assemblers like hi-fi SM, improved phased assembler from PacBio, HiCanoe. And now our compute time is down to hours instead of days or weeks, because you're going in with less data, you're going in with high quality data. And ideally you do this from a single individual. Here on the Sequel IIe instrument, we've kind of worked down the minimal amounts that we need. We can do things from 200 to 300 nanograms of DNA or more. We shear the DNA. So if your sample isn't of the highest quality, as long as it's 10 to 20 kb, so the output isn't dictated as much by the length. So a library prep today, this is how we do it in our lab. It is not how you would have to always do it. It's just what seems to work well for us. We might take a tissue sample and send it to Olga to make a Hi-C library. That's the first thing, because if we want to make a phased assembly, we want the Hi-C data and the Hi-Fi data to come from the same individual. We then grind the sample to powder in a cryogenic state we use a genogrinder with a steel bead. We do a DNA extraction by hand right now. We're using the QIAGEN MAGATRACT high molecular weight DNA kit. There are a lot of other kits out there. I'll talk about that later. And then we do the trifecta of DNA QC. So I would highly recommend, if you don't have access to all three of these approaches, that you should find somebody that can help you out. So first, and I have it listed last here, is the purity of the DNA. How many contaminants are in the DNA that you're working with? Because if there is something else in your DNA than just DNA, the polymerase on the sequencer might not work well. The library prep might not work well. You might have something that's inhibiting ligations and things like that. So purity would be taken on things like the nanodrop. We actually have a Denovix in the lab, which is like a super nanodrop that can also do qubit style readings as well. But it's a spectrophotometer based reading. So you're reading the 260, 280, 230 ratios. You can detect if there's proteins, contaminants. You can detect if there's phenol carryover, if you're doing phenol chloroform extractions. You can detect RNA potentially in the sample. What it's not going to give you a good understanding of is the amount of DNA in your sample. So these ratios might estimate that, but instead what we use is a fluorescent probe-based library quantification. That would be using something like a qubit fluorimeter, or again, we use our Denovix because it has two things. It can read the qubit and it can also act as a nanodrop. Here, you will use this number to determine the amount of DNA you have. So simply you take the quantity you have and you times it by the volume, and that's how much DNA you have to go in. If the nanodrop says there's twice as much DNA, ignore it, don't trust it, it's probably not right. DNA QC, you can do sizing. We actually use a femtopulse now, which is a pulse field capillary electrophoresis unit. But what you want to see is that there's no shear of the DNA. If your DNA is 50 kb, 60 kb, and there's no shear, great. If your DNA is 150 kb and there's no shear, that's also great. Because the first thing we're going to do is shear the DNA down to 20 kb. What you don't want to see is a big tailing smear of DNA that's making it look small. All of these things, the sequencing is expensive, right? And so if you give it to your core lab and they go and sequence it, you want to make sure you're giving them the best thing. They'll often just proceed with whatever you give them. And so you can't be mad at them if they give you 1/5 of what you were expecting if you're giving them degraded DNA, dirty DNA, whatever.
Sorry, Scott.
Yeah, go ahead.
Do you mind like commenting a little bit on the smeary part? So, if
Yep,
you're smeared,
that's going to get on
the
next slide. I have some.
Okay.
I'll have some pictures. Yeah. So yeah, what is smeary, right? And it depends too. You might have a B sample that you're never going to collect again, right? The genome size is small, so you don't need a ton of data. You might be willing to accept a worse quality sample for that because you'll never get it again and you're going to get what you get. But if it's a human cell line, there's no reason to accept something that's okay because you can go back to that cell line and try again, right? So yeah. So if it's too degraded, smear, stop. If it's too dirty, there's a lot of ways you can clean the DNA up further from just the high molecular weight extraction kit. One thing I'll just say, yeah, go ahead.
Since you're going to talk about more smeary, I guess I wanted to, also talk about too dirty. How often you have to do, like, because right, the kits are kind of doing, presumably, some heavy lifting across a wide range of
some are organic. If you're using an organic extraction, often there's solvent carryover, right? So that's one type of dirty. If you're not using RNA clean,
I just, like percent wise, how often in practice do you have to do secondary cleanups? I'm curious, from practice across the wide
range. And there's
range,
other things too. So when you're shearing the DNA, you have to have that DNA in a certain volume. And so one thing we do when we shear the DNA is we usually do an ampere concentration. So we'll take ampere beads after the DNA is extracted and reduce the volume down to the concentration or the volume that we need for shearing. So that in itself is another cleanup of the DNA. We also use, you can write this down if you want to, Millipore Amicon Ultra protein purification columns. Those work very well for DNA cleanup. So effectively, you would put your sample in there, fill it with water, spin it through, fill it with water, spin it through, fill it with water, spin it down to the dead stop. You seem to lose very little DNA. You get no degradation of the DNA, and it's quick. The downside there is those columns themselves are $10 to $15. So, we usually do a purity ratio first, and if we see we need to do it, we'll do that. Or somebody will try another ampere or something. The amperes, you always get a little sample loss. So yeah. So then we would normally shear the DNA. We have these things called a megaruptor. It's essentially just a syringe, and you go in and out with the syringe, but the machine does it for you and does it at very specific speeds. If you don't have one of these machines, you can find a needle. You can put your DNA in a syringe and go in and out of the needle. You might have to play with it a little bit, but there are published protocols of just using needles. The nice thing here is it does like 8 in parallel, or now it can do 12 in parallel. So if you're doing a lot in a day. So you shear the DNA down to your 10 to 20 kb size. You put on these SmartMill adapters. The polymerase binds to the adapter. And then this becomes a circle. And so what you're doing is you're reading off these reads over and over and over again. So you get the positive and negative strand of the read, which is then used to create a consensus accuracy, a consensus read with very high accuracy. So even nowadays, most people aren't doing size selection, but if you were doing a standard sample selection, sample size, you might have micrograms of DNA going into this. You could size select it on something called a Sage Pippin or Sage ELF, blue Pippin, those kinds of things. They're basically an electrophoresis unit where you can collect off of that. We don't do that because the Pippin always eats our DNA. So if we put in so much amount of DNA, we get back a smaller fraction of that. And then we load and run on a SQL 2 system. So it's a molar loading. So depending on the length of your DNA, is going to indicate how many nanograms of DNA you need for the loading. So if we know the sample is small in physical size, we may shear it a little bit more because it's going to raise the molar loading of the sample, because shorter fragments are going to have a higher molarity than longer fragments. So that's a little trick where like you can shear it maybe down to 10 kb and still get pretty good data from something that you might only have hundreds of nanograms going in. If you have micrograms, you might as well shoot for 15, 20 kb because you're going to have the loading molarity you need. If you underload a SQL system, the yield will be heavily impacted. Is there a question real quick?
I want to, don't want to take over, but I guess I'm the one who has most.
I love interactive. Yeah.
I wanted to, so basically you're saying that, size selection is so lossy that it's like it's pretty much never worth it, even as a compensation for, right, like the smear or degradation. Basically,
so
my question is,
in those cases, we just never have those samples. We never have those 10 microgram
practice,
samples.
Right? If you guys are doing loads of this and in practice you never do this, that probably means that that's just not, it's not a good strategy. My question was, right, like basically, right, imagine a case where you have, you know, like a bad sample, but you have loads of it, right? So you could do the, you know, smear,
Yep.
but then just do size selection for the longer things. But it seems that like that's basically, you know, like not a good strategy. At least it
Right.
doesn't seem
And
like you in
it's
practice ever do this.
and it's a little bit crap in, crap out too. So if the DNA is sheared, it probably also has a lot of nicks in the DNA, probably has a lot of damage in the DNA. During the library prep steps, there are steps to
So like trying
repair
to
the
save it
DNA.
with like size selection is just not going to
Right,
do the
but
trick,
if you're going to
probably.
have shorter DNA, but maybe it's damaged throughout, so then the polymerase
Mhm.
isn't going to be able to go around. So then the yield of that run might be lower. So you know, then the question will be, what can we get a better sample coming in, right? And it all depends. If you're working in the endangered or extinct organism, you're willing to accept lower quality than if you're working on a, I don't know, something like
And
an
does
ant
and
out of
and
a
does
colony.
and does SQL do the same thing? Like, you know, there is like bias towards shorter pieces. I guess, you know, in practice it has to be, but basically, in addition to just the fact that, you know, there are spots that, you know, can be occupied by shorter things, is there like some kind of inherent systematic bias towards shorter because of some, you know, they like amplify faster, like things like that?
Or they land faster, I think. So if you have, I won't go back, but
So you will get disproportional bias towards shorter
short
things,
stuff
even if your library just contains
has
like
long
relatively.
chains.
Okay.
Yeah. So
Got it.
the 8M cell has 8 million little holes that the DNA can go into. If you have a lot of short stuff, the short stuff's going to go in the little holes before the long stuff, right? So at the very end of library prep, they do have a size selection method that uses ampere beads. It's like a permissive ampere that cuts off the DNA around 5 kb, and you basically can remove those sort of 5 kb fragments. But if you have something sheared from 3 kb up to 40 kb and it just looks like garbage, you know, to pip in it, you know, I would probably say there's something wrong with that sample. So this is an example of what you want to see as a good. This is actually on our old fragment analyzer. I didn't update these slides. Basically, that tops out at 50 kb here. You can see most of the DNA is bigger than 50 kb. And then we would shear that DNA using the megaruptor. So this sheared to 17,578. You see a little bit of shouldering here, but it's nothing too bad. And then the final library, like I said, has this ampere step that removes fragments lower than 5 kb. So you can see a lot of that 3 kb comes out, and you're basically matching the size of the shear to the final library. If you shear it and your final library is substantially shorter, then you'd say, what happened during library prep to make that library shorter, right? Again, this is a lot of the things that happens at the core lab, but you can do this in your own lab too if you want to. But getting feedback on this, if your run is bad, what's the shear look like? What's the final library look like? What did the, you know, getting that feedback can really help in troubleshooting why something went wrong. So like I said, this would be the blue pippin that could do the size selection. We don't use that. In doing so, we don't get sample loss in our samples. If I was working with huge amounts of DNA, I might be convinced otherwise, but I think there are ways around that just by having really good sample input, really good DNA extractions, and that kind of thing. So then you sequence this thing on the sequencer. These are some older stats, but still pretty good. So remember, we're going around the circle over and over and over and over and over again. So it could generate between 2 and 4 or 5 100 gigabases of data. But because you're going around the circle, you're not getting all that data back. You're only getting one representation of your, what they call the subread for all those times around this circle. So the difference between total gigabases and unique molecular yield is the total bases are all the times around the circle. The unique molecular yield is the one representative insert of that circle. So here you can see we're getting about 29 gigs, 50 gigs in this case. And the N50 again is almost matching what we saw on our little chart. It was 17,578. Now we're getting mean of 17,384. All sounds pretty good. One run is probably sufficient for 500 to 1 gigabase genome. Library yield may allow for multiple cells as well. So you might make a library and you could run this on multiple cells over and over again. The Revio system that's coming out will increase that dramatically. So they're suggesting a single run. So the reason the Revio system exists is because everything's circled around humans. Human genomes 3 point whatever gigabases in size. Right now on this equal 2e, you have to run multiple cells of this equal 2e to do with human genome. On the Revio system, it's one cell, one human genome. So it simplifies things. It runs in 24 hours, and that's kind of where
Scott,
that goes.
and the expectation is that, right, like one library, one cell. So
One
there's no
library, one
complexity
cell.
issues of any sorts.
Yeah, in our case, though, if we're doing tiny little B genomes that are 300 megabase, now we could put, 5, 6, 7, 8 of those on one cell and potentially get all the genomes back.
Do they have like a demultiplex option?
They have
of sorts?
barcodes. Yeah, they have
The
about,
barcodes?
I think, 16 barcodes that you could, essentially when you ligate on your adapter, that contains it's a very long barcode. You do get a little loss, and then you also have, it's not as simple as indexing in Illumina sequencing, because Illumina, all your libraries are the exact same size. They're all, let's say you made 300 base pair library, 400 base pair library. Here, your libraries could be of different sizes because of different qualities, different shearing parameters. So if you're trying to do equal molar pooling, it can be very hard to dial in equal molar pooling on things that are like shear smears, right? So, they say these things, but in practice, I still would probably just sequence one B, one cell, because the price of the cell is cheap enough that we can afford that. But if, you might get something that is not completely even between all your samples. And then
I never had to deal with demultiplex. Is this like automatic on
it can happen
anything
on
PacBio?
instrument? Yeah.
Okay.
So you just get reads back. The naming convention's a little different, but it's all on an instrument. So simply, where I'm up to at this point, we take the sample in, PacBio Hi-Fi library prep sequencing, Hi-C library prep, Illumina paired in sequencing, and then we kind of go through this assembly process. So we kind of clean the reads, do a contig assembly. You guys are all experts now on scaffold assemblies using Hi-C data. We then do some steps to confirm that this is what the genome should look like, that the genome looks complete, and that there are no contaminants in the genome. The outputs of these, so this would be a K-mer analysis that would say the genome is in a haploid state, and this is the genome size it should be. We pull out the mitochondrial genome. There's a whole mito Hi-Fi pipeline for pulling mitochondrial genomes out using Hi-Fi tools. We might do repeat finding to sort of characterize the heterochromatic regions of the genome. BUSCO is a universal single copy ortholog database, so you can see how many of the genes that are supposed to be in your genome are there, and it's a very good assessment of completeness. And then blasting against NCBI databases for things that are clearly identified as not your target. So if we're working with bees, they often are carrying pollen. We'll get many, many reads that look like alfalfa from the alfalfa leafcutter bee, right? Or whatever. And we want to remove those. We don't want those labeled as bee and NCBI. So then we filter that all down and import into NCBI. So I kind of went through this. There's also an ultra low input that I'm going to talk about in a second. But the assembly process, we run this Hi-Fi adapter filter. You could run other programs. There are a lot out there. We contig assembly with Hi-Fi assemb. We use a YAS tool. Oh no, I'm messed up here. I scrolled. That's the worst. We use YAS, but there's other tools for doing the scaffolding, and then we do manual curation in the amazing JuiceBox assembly toolkit that Olga's group has developed. Like I said, we do some of these analyses for pulling out things, for ensuring things look right. Blob Toolkit is a visualization tool for things that should be in your genome, like both BUSCOs, as well as things that you don't want in your genome, like contaminants, microbial things, and things like that. And then it gets filtered and finalized and submitted. So the timing of this, in the past, this has been a long time. This is our only reference to locust in this talk. Did you see the hopping? We all love the hopping. Yeah. So somebody sends us a sample. We can, in a week, sequence that library, prepare it and sequence it. In a couple days, we can go through the assembly process. In a week, or I don't know how long you guys take to make a Hi-C library. I might be over or understating that. We can, prepare a Hi-C library, sequence it. So in 2 weeks, you can be at the scaffold assembly step. this is somebody just kind of doing it on their own, not in like a production sequencing kind of thing. Maybe you'll take a couple weeks to QC on that data running some of these tools. You filter it in a day, decide what you want to submit, and then you submit it to the public repository. So after you submit it to NCBI, you might give them a month before that data is actually going to be public. So really from start to end, you might be able to look at, in a best case scenario, a month to 2 months, and a worst case scenario, maybe 4 months to get your data public in NCBI and completely done. So it's pretty quick. It's pretty straightforward, and we have developed pipelines for this. So I'm briefly just going to go through some examples. This is an Indian meal moth, a tiny little moth. That's with the 13.5 kb shear. We got 12.8 kilobase subbreed, so very close to that. 52 gigabases of unique molecular yield. And this essentially assembled into, so Lepidoptera have 32 chromosomes, and 90% of the genome is in 35 pieces at a contig level. So there were very few contigs that had to be joined to build this into a chromosome scale assembly, and then the Hi-C data looks very beautiful. This is just a different representation of the Hi-C data at a different filtering scale. So that worked really great. That's a little pest moth, not too big, only 291 megabases. These are some Diorhabda beetles that are used for biocontrol. Some of these, we actually sequenced 2 of them on a single cell. And again, we're getting these N50 contig assemblies that are not much different than the number of chromosomes we expect to be there. In this case, Hi-Fi ASM was used. In this case, Hi-Canoe was used for the assemblies. This is where we're trying to push the limits of low input yields or low input DNA into the sequencer. So these are these tiny little pest beetles. If you've ever gotten rice at home, you might be culturing some right now in your house. They are pests of grains, stored grains, and things like that. And they are on the order of millimeter or sub-millimeter in size. So again, we're only getting hundreds of contigs, very good contig assemblies, and things like that. Is it going to go? There we go. Some more recent things. This slide's a little bit old because we have a lot more coming out on this. But this is low input Hi-Fi assemblies that just have extremely high contig N50s. And so this is the contig space for these, color coded by the chromosomes. And so you can see this entire 3L arm. This is a chromosome. This 3L wrote in reference to Drosophila, but this is a whole chromosome is a single piece. This is 2 pieces. This is 1 piece for chromosomes that are on the scale of 60 or 80 megabases in length. So yeah, it's pretty amazing what's coming out of these. In addition to having many samples below the standard inputs, sometimes you get things that are very, very tiny. Like we can only get like 100 nanograms of DNA or less. Beetles, thrips, whiteflies, mites, mealybugs, whatever. So there's actually an amplification-based ultra-low technology that PacBio has. It's based off of long-range amplification-based kits. You can input at the 5 to 20 nanogram level. And so you basically have 5 to 20 nanograms of DNA. You shear it, which is funny because you can't even see this DNA. And so you're hoping it shears to 10 kb. Lay it on PCR adapters and do a PCR step to amplify the library. You're going to get some bias here because the whole genome's not necessarily going to amplify using polymerase. So it's just going to be hard regions based off of GC content, repetitiveness, things like that. And then you pull these PCR products. So you're doing sequential amplifications and size select that library and sequence it. We tried this on one of these tiny little beetles that we just couldn't get enough to run a thing on. This is a sawtooth grain beetle, everybody's favorite tiny beetle. What's interesting is it's not short, it's just flat. So we didn't get very much DNA from it. And so the standard input kind of led to this very poor assembly. So the N50 was below 100 kb. The Buscos had a degree of missing, but also a degree of duplication. So it just wasn't able to assemble well. So we start with our 5 nanograms. We bump the N50 up above a megabase, so it's 1.8 megabases in size. Again, this is a very tiny genome though, too. So, if you're trying to do this on something with a 4 gigabase genome, it'd be a huge uphill battle. But we're complete in the score skyrockets up, where we have single copy complete at 99.4% or 96%, only 2% duplicated, and 99.4% of the complete BOOSCOs are present. So if nothing else, we have a complete representation of what this genome should be. So we're do this for other ones. It is beneficial to do this over pooling individuals, because all of these new assemblers are based off of diploid assembly, assuming haplotypes and things like that. How am I doing on time? All right, we'll start cruising. So we make pretty good progress on single insect assemblies of low input. Recent hi-fi approaches yield near chromosome contig assemblies from nanogram amounts of DNA, which also simplifies a lot of the high C assembly sides. You're not having to do as much editing. I think the assemblies just run faster. We're only doing a handful of contig, things like that. We're coupling that with high C for scaffolding and validation. And we've done lots of these over well over 100. This is a representation that we do use for internal to look at for our agency, just saying we're covering all the USDA ARS agency locations and their pests, insects, and things like that. This is how the 6 locusts that were done as part of the BPRI grant, 1 old world and 5 new world. And these are huge genomes with 3 times the size of the human. Each chromosome of a locust is larger than the entire Drosophila and the melanogaster genome. So we're talking, these chromosomes are in excess of 100 megabase easy. We're also using this approach for genome. So we have our genome that lives on top of our sequencer and tells everybody they're doing a great job. We've sequenced about 100 native bees in fiscal year 2022. Going into 2023, our 2023 should target for another 100, and also doing some honeybee pangenomics. So these are native bees to North America. So I just want to talk briefly about some of the futures of sample prep and assembly, or alternative ways you can do things. Alternative sample and extraction techniques exist. New library prep technologies exist. There are also alternative sequencing approaches. Telomere to telomere is kind of a jargony term right now that everybody needs to do a telomere to telomere assembly, and there are some sort of prescriptions for doing so. So I mentioned we use the MagaTrack high molecular weight DNA kit. There are a lot of other kits out there. I'm not for or against any one of these kits. The best kit is the kit that works for you, which might be different for different people. We got the Kaijin kit. New England Biolabs has a high molecular weight kit. BioNano, or sorry, not BioNano, Circulomics, which is also actually acquired by PacBio, has a library preparation method. There's this Monkey Fire kit out of the UK. And then there's old school methods, agarose plugs, phenylchloroform extractions, doing organic extractions and things can make very, very good DNA. If you can compare these, this table's from the NCBI website, so NCBI is highlighted, or NEB, not NCBI, New England Biolabs is highlighted. This is not an endorsement of their product, but it just, to me, this kind of shows they're all pretty much the same. So I don't know why it says here the Kaijin MagaTrack DNA is heavily shared. We don't have that problem, but of course, New England Biolabs wants to say they have that problem, and that's why you should use their kit. So again, there's a lot of it has to do with how much input you have. The MagaTrack for us is very good at scaling down to low amounts. I know a lot of people in plant genomics, like the Circulomics nanobind kits, or still favor phenylchloroform reactions, things like that. This company that exists for FFPE, formaldehyde fixed paraffin embedded samples, Purigen, was recently acquired by BioNano Genomics. They use ionic purification method. It's an isotaco electrophoresis method. So it's not isolating based off of size, but based off of thing. So it actually can purify DNA very well. We've demoed this or done a thing with the company. I'm not specifically focused at high molecular weight DNA, but now through their acquisition by BioNano, which is a optical mapping technology that needs like megabase scale DNA, they're pushing towards changing their system for also working for high molecular weight approaches. So these things could exist and make things easier. Again, the cost for this is exceedingly higher than what it would be just to do an extraction by hand. On the library preparation side, you're normally fragmenting the DNA, doing damage repair, end repair, ligation, purifying, primer annealing, binding the polymerase, and then you get to sequence. So it's a lot of steps. The new Revio sequencer can sequence 4 cells a day, 24 cells a week. So that's a lot of extraction, a lot of library prep and things like that. So some technologies are coming out to automate this process. This is a Miro Camavis from Miraculous. It's a Lecter wetting device that moves little dots around and can automate purification of a PacBio library. The downside is it's 1 cartridge, 1 library. So if you want to run 4 libraries at one time, you need to run 4 of these machines independently to make those libraries. Oxford Nanopore similarly has their Voltrax system. Again, it's a 1 library at a time kind of thing. These are a little more niche-y, kind of cute, I guess. And then, it's going into the old school liquid handling companies. So this is, TCAN has this toaster, I forget what it's called. It's for Illumina library prep, but probably, down the line they could adapt for high molecular weight. It's cheap. And it basically is a mini liquid handler that just does 8 preps at one time. Or if you have on site a biomech or TCAN or whatever, you could automate the preps yourself. Any questions on those things? This is me just kind of throwing out random technologies that haven't been sort of proven out there.
I've got a quick question, Scott.
Sure.
What would you recommend for like a very advanced high school class?
Very advanced high school class, do it by hand.
Okay.
I don't know, why not? You need a center. So the other thing, if you don't have a shearing device, I said you can use a needle. The other thing you can use is a Covaris G tube, which is a little spinny guy. You just need a centrifuge to run it, and it does a pretty good job of doing it. I guess you need to do it by hand, and a lot of, none of this stuff is too cheap. But if you got the right funder, go for it, right? Nanopores may be a little more approachable, and the way their kits are present, you could do a rapid kit, which is very quick, only requires a thermocycler. It's using a transposon-based insertion for putting in the stuff.
That's
Some of
what
these
we're
other
thinking,
things just take a while. So if you're trying to do it in a classroom. So the sequencing technologies, this is not to scale. The machines are not getting physically larger each year. We're not crushing. We're eventually going to have something the size of the Empire State Building, but they are producing more data. So we went from 150,000 little sequencing sites to 1 million to 8 million. So now 25 million per cell, but now also these all ran 1 at a time. This can run 4 concurrently. So this is going to dramatically increase the output. So you could sequence a whole locust genome in a day on something like this. So this is their Revio system. They have their cute little branding. Give me a thumbs up or thumbs down if you like the look of that machine. Does that look like the Death Star? If so, is that cool to you? Or do you think it's kind of ugly? I don't know about their purple. You guys like it? I don't know if I like it. It's a little scary. It doesn't look approachable. We of course have Promethean out there, totally another way. Nothing wrong with this. You can generate lots and lots of data. So it looks like this. Secretly, you have to hide this giant box underneath the machine. This is a large Promethean. They are coming out with a smaller Promethean. If you're doing whole genome sequencing in eukaryotes, you really want to be working on a Promethean system and not as much the MinION or GridION systems. So they have these style flow cells. They have about 10,000 pores or 2,600 or so sequencing things can happen at once. And they're I don't know who names this stuff. Their current flow cell is the 10.4.5 flow cell with V14 chemistry. Why they just don't call it the V14 flow cell to match it with the chemistry, I don't know. And it gives Q20 accuracy. So this is a distribution of what the reads would look like from this. We have a machine in our house. This is what our data looks like as well. You can maybe get 100 to 200 gigs of data off of the machine, which is going to be comparable to the Revio system. The cost for that is about 50% more than the Revio system. But what they have come out with too is what you do when you nanopore sequence something is you unzip the DNA and one strand goes into the pore and gets sequenced. So what they've done is try to increase the probability that the next thing sequenced is the other strand of that same DNA. So they got sued for coming out with a technology that would just barbell one end and sequence the two strands concurrently by PacBio because it was too similar to circular consensus sequence. Because this is not relying on that, it's just relying on proximity to the pore, you're essentially getting two reads happening at once. And instead of going around the circle 10 times to consensus something, you're getting two reads that you can now consensus. And so that can push the accuracy up a little bit above Q30. Not all of the reads in the run are able to get this. Both reads happening, it kind of happens not randomly, but it happens at some proportion. And really the technology here is now pushed at increasing the proportion of that happening, maybe from 30% to 70 or 80%, so that your standard read coming out could be Q30. Otherwise, the data coming off of Promethean is significantly lower accuracy than a PacBio sequencer. Not to say, but it also the reads can be significantly longer. Any questions on that real quick? So sequencing technologies for short read sequencing used to be only Illumina, right? So you were as good as whatever model Illumina sequencer as you had, going from your iSeq, MiniSeq, MySeqs up to the production scale sequencers. In the last year, Illumina has had a little competition in the system. BGI-seq has their sequencing technology that's based off of complete genomics. Really, the two Illumina sequencers that are going to be sort of stand the test is this NovaSeq X series, and then smaller labs may have a NextSeq 1000 or 2000 for benchtop sequencing. Ultima Genomics is sequencing on this giant wafer, offers a $100 human genome. While the NovaSeq X series, when they come out with their latest and greatest chemistry, is probably going to be at least twice that expensive. So this is a singular genomics G4 system. This guy is focused on sample batching, so it has 16 independently deliverable flow cells. It sequences at above probably $10 per gigabase. So the best price you're going to see on a NovaSeq X is $5 to $10 probably per gigabase, but you're not having to batch all your samples together. So the idea here is you could run little things all at once. This is a PacBio Onso system. This is sequencing by binding. So they're claiming that this will have quality scores up in Q40 or even Q50 when it's released. Its release is probably going to be mid this year. These other ones are sort of available. Ultima maybe not. We in-house, we actually purchased a VT system from Element Biosciences. The big thing here, it's similar in size and output to a NextSeq 2000. It has two complete independent sequencers and sequences at $5 to $6 per gigabase. So we can get in on that NovaSeq level pricing, but on an in-house sequencer. The data quality seems to be very high as well. They're on their first chemistry release, and the turnaround time's 48 hours for a sequencing run at 2x150. So there's a lot of different things out there. So if you're doing a project, you know, whatever you have locally, you might look at, but at times sequencing on this might cost as much as $15 to $20 per gigabase, whereas sending it to NovaSeq X might cost 10 to 5 to 6 to 7 to 8. This is $6 per gigabase, you know, so you can get a lot more bang for your buck. But, you know, some of these are going to survive and some probably are going to die. So we'll see. There's also different assembly approaches. I got about 5 minutes left here. So, you know, the prescription that we presented here, this Hi-Fi plus Hi-C, the next thing that people want to do is Hi-Fi plus nanopore, ultra long reads. So both the Hi-Fi ASM algorithm can integrate ultra long nanopore reads to improve the assembly. You just add it in as a thing, or the Verco assembler, which is from the Canoe group. The prescription there is to have, you know, in both of these would be about 30 to 50x of Hi-Fi data, and then 100x of nanopore data, but specifically nanopore data that's longer than 100 KB. But what it's able to do is basically phase and create telomere to telomere chromosomes for your critter. That's great if you're working with a whale or something and you have unlimited amounts of DNA. I don't know how applicable this will be to insect world because all the material has to come from the same individual. One of the big caveats here is even using their ultra long sequencing approaches for Oxford nanopore, only a small proportion of the reads are greater than 100 kb in length. This is their best. This is what they have on their website, right? So this isn't running one Promethean cell. This is running many Promethean cells, throwing away all the data shorter than 100 kb and only using the data longer than 100 kb to go into your assembly. That's going to depend on sample quality. That's where your extraction method is going to matter. That's going to matter how much DNA you have going into it. How many small things, right? Olga said those small things get sucked up by nanopore immediately, right? You got to get rid of all the smalls. So, you know, in a production scale, how is this going to come out? I think most of it's going to have to be modifications or improvements to the sample processing process to increase this. It may be too that you don't need 100 kb reads every time, because maybe this is really about achieving reads that go beyond the repeat structure of the genome, right? And so if you're doing insects, maybe that's smaller than what you would see in vertebrates and things like that. This has really been only tested in a lot of human samples and in some vertebrate samples and things like that. I was going to talk about what we do with this, but I'm out of time. So I'm just going to slide through pretty pictures. Look at those assemblies. Look at that. Chromosomes, inversions, translocations. Beautiful. Look at that, a little inversion, a little flip, a little autosome onto the Y chromosome. Beautiful. What a great presentation that was. Sorry, I ran out of time for that. But I do want to say just generally our location has expertise in insect species strain detections, diagnostics, specialty crops, microbiology, sterile insect techniques, and all of the instrumentation that I talked about today we have in-house. So genomics, high performance computing, bioinformatics, and things like that. This is our lab folks in front of all of the machines. The Hawaiian shirt is optional, but this is part of the group right now, all smiling. This is how they really feel every day. And I'd be glad to take any questions. Also, too, if you are graduating or whatever, we always have positions at a post-master's level or postdoctoral level. And there's also several vacant positions that'll be coming through for permanent scientist positions at our location. So if you're interested in those things, please reach out to me. Any questions?
Thank you, Scott. You have put so much effort to make a very nice, like figures, everything. I think it's take too much time.
I used a lot of things that already existed in a different context. Yeah.
Okay, anyway, thank you very much.
No problem.
I will leave to the participant to have few questions. So we have 4 or 5 minutes.
And I'm happy to, if you're approaching a sequencing project, feel free, you can reach out to me with no obligation or expectation just to ask questions, because the worst thing is taking grant money and then it not working out because of something very small that could have been changed, during the collection process or the processing, that makes things the difference between it being beautiful or being a failure.
So everybody's being quiet, so I'm going to butt in. If we were to design population sequencing of locusts in Africa for a future BPRI grant, what advice would you give us?
Yeah, I guess what is your population question? Is that a population genetic question? Is that an evolutionary question about like maybe regions of the genome under selection because of something? How are populations changing between these things, you know? You know what I mean?
I
I don't
was
know too much
thinking, what
about
is the population genetics of a swarm?
like
You
how
know?
what's the variation? I would just
What's the
do
before and during and after? And
Yeah.
so then you have this issue that it's not in the US, you have the shipping issue. It's not clear to me as if it is easier to get the samples here or to send the machines there.
Yeah, so we actually had really good, whoever, I forget the name, who sent us samples, it worked out very well, but that was from one location, the easiest location, right? But I would just go back to your short read sequencing for those types of things if you're just looking for markers. You know, long read sequencing sounds good. But if you're not looking for structural changes, if you're not looking at phasing, if you're not looking at those things, you don't really need it. You could even do a reduced representation library if you just want to say, you know, what is the structure? What is the population structure? What's a DAPC type of analysis look like for these populations and things? So, yeah. Because the other thing, I mean, you might be able to get the samples and the money to do it. Even with the Revio system, it's still like 4 cells to do a locust at least, you know. Or just be, you know, it's a lot of resources. Yeah, and could you get, you know, normally, so we do, I didn't talk too much, but we do population genomics in differentiated fruit flies, where it's kind of a boundary between population genetics, but also hybrid species complexes. So there's like evolution happening and things like that. And our data sets usually are in the order of 100 to 500 individuals across things. And, you know, I don't know that locust you could generate that comprehensive of a data set. I mean, you get 500 locusts, they're all just going to be from the same swarm, from the same location. Not hard to get 5 locusts, hard to get 500 different locusts, right?
Yeah, that's the thing that's well, maybe we need to talk internally, but that's the kind of question I think might be the harder bit.
When again, back to this, that right, we've discussed to do some population analysis using short read sequencing, given that much of it seems to be around like huge inversions and like nested inversions. That's easy to see. It does not have to be extremely deep because they're so big, et cetera, et cetera. So just back to that discussion.
Yeah. And even high C or something can validate that as in the same way the long reads could, right? Or even the short read.
That's what when I say short reads, I mean Illumina high C. Yeah, that's exactly what I mean.
So it all depends if you can get what kind of samples can you get for the samples, right? The other thing we see in our flies is somebody will say, oh, I caught this fresh, and it's totally, we can tell it's a fly that's been dead for weeks sitting on someone's desk, or, you know, so they can't get anything from it but bacteria.
Yeah.
Is there any other question? If there's no question, I'm very thankful for Scott for doing that for BPRI. Thank you very much for your time. And I will share this recording with all students in BPRI. I know that few people cannot attend due to the course they are taking. So I apologize for low attendance today because some people are really busy this day and some have spring break. So hopefully I will be able to share every recordings with them. If they have question, I will say that please send an email to Scott. He's happy to answer.
Yeah. Or get on a plane. You can fly out here and we'll talk in person. No shame in that. All right, guys. Thank you so much. Really appreciate the invite.
Okay, thank you. Bye-bye.